Hey guys, welcome to the second part of Chapter 7. In this topic, we're going to talk all about translation, which is the RNA to protein. And then we're going to wrap up a little bit with talking about some origins of life because you haven't had enough of it yet. So we're going to make sure we tack that in here too. So make sure you understand all the aspects of this topic as well as how it relates to topic 1 because these two very much go hand in hand with each other. So as I said, for today, this topic outline, we're going to talk about RNA to protein, which means we have to talk about the codon system, tRNAs, the ribosome again. We're going to really break down the structure of the ribosome. Talk about the process of translation, polyribosomes, translation antibiotics, and the degradation of proteins. We talked about how RNA is degraded in the last topic. In this topic, we're going to talk about how proteins are degraded. Then we're going to wrap up by talking about RNA and the origins of life with catalysis, the storage of information, nucleic acid evolution. As always, here's our topic objectives. Now, I know there's only a few here, so don't be scared. Don't be scared, but don't take them to be simple. They have a lot of rules or a lot of layers in them. So if you need to talk about any of these, please let me know so we can work through those. So as I said, the first thing we need to do is we need to talk about the codon system. This is how we get from RNA to protein. And every codon is specific for a specific amino acid. The first two nucleotides are the most important, and you can see that here. Like if you look at alanine, the alanine will always start with a G and a C, whereas that last spot in the codon, because it's a three base code, that last spot's known as the wobble, and that can change, which means that it allows a little bit of flexibility if there's a coding mistake. That allows for that. But the 3 nucleotides equal 1 codon. So it's very important to understand that. And this is where we talk about frame shift issues, and we'll talk about all of those issues in the next chapters, or in the future chapters. But it's really important to understand 3 nucleotides equals 1 codon, and the last spot, always the third spot, it's not just a random 2, but always the third spot in the codon is a wobble nucleotide, so it can change and still code for the same nucleotide, or same codon, the same amino acid. So it's really important that we understand that and the reading frame aspect of it. So why do codons matter? It's because they're read by the tRNA. tRNA has an anticodon on the bottom of it. You can see that here in the blue on the structure of the tRNA. This anticodon is the complementary sequence. So it's really important that you understand that. It's just like when we talked about DNA replication with complementary sequences, and in this case it's the same way. That anticodon always codes for the same codon. The same thing's true for the other side of this, of a specific tRNA, and that will always code for the same amino acid. And it actually is checked when these amino acids are attached to the tRNA. They're checked for their anticodon to make sure that there isn't a mistake. So this is just as specific as we talk about any other enzyme or antibody or any of those proteins. tRNAs are very specific for their amino acids. So how do these amino acids get onto the tRNA in the first place? They use amino acyl tRNA synthetases. That's a mouthful, but it's an enzyme. And what this enzyme does, as you can see in the image here, is it has the amino acid and the pocket for the amino acid, a pocket for the tRNA. They will come into the ligand binding sites. The enzyme will check to make sure that anticodon is appropriate for the amino acid that's going to add to it. Then using will create a high energy bond between the amino acid and the tRNA. Then it will release it from the enzyme complex, and it will go out and join to the rRNA sites and allow for the transfer of this amino acid to the growing primary structure of the protein. So let's talk about that ribosome now and the rRNA. Remember, the ribosome is a combination of proteins and RNA, making it a ribozyme. So it's still an enzyme, but it has ribosome, it has a RNA catalytic sites. Remember, it has those 3 catalytic sites, and we'll look at those in just a second. So prokaryotes and eukaryotes have 2 different types of ribosomes. The eukaryotic ribosome is 80S, which means that it's a larger protein or a larger enzyme than the prokaryotes, which is the 70S. Now it is composed of 2 parts, the large subunit and the small subunit. And both of these have RNA and proteins in them. These come together to form almost a sandwich. They look like a little, like one of those little guys from the Super Mario Brothers. And they will sit on the mRNA to read it and provide the catalytic sites for the tRNAs to come into. So it's important to understand the structure of the ribosome and how there are different components. It's not just that the ribosome is rRNA, it's that the rRNA is part of the ribosome as well as proteins. So now let's look at the process of translation. So what we have here, and you can see through all these images, is we have a whole bunch of steps. So the first thing that happens is the mRNA becomes present, and we have an initiator tRNA, which is always a MET. This will sit with the small subunit on the appropriate starting region of the mRNA. And you can see that here with the AUG. This is always the start codon. Once that binds, there's a signal that the large ribosome needs to come in and bind to the small subunit. And at this point, now we can talk about the translation that we've seen before. This is where the next tRNA will bind into the A site. Now we have, and in this image, you can see we have a MET with the next amino acid. In this case, it's just a generic amino acid. What will happen then is that MET group is transferred to the second amino acid, and the initiator mRNA, or the initiator tRNA, is going to move to the next site, and everything's going to shuffle down one step. And you can see that here in the big picture in the middle, how this works. So we have a tRNA that's in the P site transfers its amino acid to the tRNA that's in the A site. Then we'll move to the P site tRNA will move to the E site to exit. The tRNA that's in the A site will move to the P site, and the process continues. You can see how this works. Now this keeps going until a stop codon is read. When the stop codon is read, we have a specific protein that will come in, sit on the code, and this will signal for dissociation of the ribosome complex. And you can see how that works here. That's that big red release factor that binds. So make sure you follow through how this works and make sure you understand that. I have some videos up for you in Blackboard, and I encourage you to watch those because this process makes a little bit more sense when you can see it in motion. Now we need a lot of proteins within the cell, and we need a lot of the same protein. And it would be really cumbersome to have one complex making the same one over and over again. So what we see is polyribosomes. This is where one ribosome will hop on and will start making it, and as soon as it moves far enough down along the line, another one will hop on, and another one will hop on, another one will hop on. And this in prokaryotes will actually happen as the mRNA is being synthesized. And this makes sense that it can happen that way because in prokaryotes they don't have a nucleus. So the mRNA is being synthesized right where the ribosomes are as well, as opposed in the eukaryote, it has to be processed and moved out of the nucleus before the ribosomes can do that. Now, translation is a really interesting place to look at for drug targets, especially antibiotics. If we can stop proteins from being synthesized, we can essentially halt cell cycles because if they can't make proteins, they're not going to be able to do what they need to do. And so this is a really interesting area, and you can see a couple examples of some antibiotics here. Now, it's really important that when we talk about antibiotics, the antibiotics are targeting specific areas. Now, in the prokaryotes, if we're going to target a bacterial infection, for instance, we just need to make sure we target something that is specific to a prokaryotic translation process, not a eukaryotic translation process. And a lot of the times we'll get these from natural products, such as fungi or actinomyces or something like that. But there are some that we can use to target eukaryotics, but we have to be even more specific that we're targeting something that is specific to that type of eukaryotic infection. And that's why eukaryotic infections are much harder for us to treat than bacterial infections. So just be aware of how this works. I'm not going to ask you to tell me what these different antibiotics do and where they target. I just want you to understand that concept of drug discovery. Now let's talk about how we degrade proteins. Proteins get degraded for a variety of reasons. Either they're old, they're misfolded, we don't need them anymore. There's a variety of things. And one of the main processes used in the cell is the ubiquitin proteasomal system. In this process, proteins are tagged with a specific little molecule known as a ubiquitin. And this ubiquitin is now a tag that says, I am ready for destruction. And that what happens is this will help funnel the proteins to what is known as the proteasome. And you can see that here with this barrel complex. This is the proteasome. The proteins will go in, and there's a bunch of enzymes in there that are going to break down those proteins into amino acids. And that allows for the amino acids to be recycled and for the proteins to no longer be out in the cell, possibly causing damage or worse, just wasting resources. So let's review transcription and translation real quick. because this is the heart of this whole thing. And so what you see here is we start with our DNA code, which is full of introns and exons. We have the initiation and transcription using the transcription factors. Remember, there's either the Tata box in eukaryotes, which is what we have an example of here because we have a nucleus, or we have the promoter regions in prokaryotes. This is then transcribed into mRNA, and then that mRNA has to have its cap, the 5' cap, the poly A tail, and must be spliced to remove the introns that are not needed before it can be exported from the nucleus. And as it is exported, it's checked on. Remember, it's a selective permeable membrane in these nuclear pores. Then once we're out into the cytoplasm, the mRNA can either be degraded if it needs to be, or the initiation of the protein synthesis will occur. This is where we have the small subunit of the ribosome will bind with an initiation tRNA, that MET, remember it has to be MET for the AUG start codon, and then the larger subunit will join and we'll go through the process of translation. And then once we're finished, we reach the end, we reach a stop codon, the release factor will come in, release the large and small subunit from the mRNA, and either that mRNA will then be degraded based on the sequence in the tail, or will be set to be remade into another protein, into the same protein again. So make sure you understand how this goes. And then once the protein has fulfilled its life, its role, and it's done, then it'll be degraded through the ubiquitin proteasomal system. So RNA is pretty cool, as you can tell. It does a lot of things, right? It can serve as the blueprint of genetic information. It can also be a catalytic, it has catalytic properties. And so a lot of people, a lot of researchers are starting to argue that RNA was actually the first nucleic acid in the start of life, and that all life began with RNA, and that DNA evolved later. And so it's a little interesting to think about it, and I just want to explore this concept a bit more. I want you to be familiar with it, but we're not going to go into too much detail. So as I said, we know that RNA can catalyze reactions. We just saw that happen. And we know that with the laws of thermodynamics that life has to have the ability to catalyze reactions, because the reactions that we need are not going to always occur in a negative delta G environment. So it's really important to understand that. And if we look at our macromolecules, we only have 2 macromolecules that have the ability to be catalytic. We have nucleic acids and we have proteins. However, proteins cannot reproduce themselves. And so that's where this process of maybe our RNA using ribosomes or ribozymes can reproduce themselves, and how they start to think that this might have been the evolution, so that the macromolecule of RNA is the only one that exhibits the properties of the life that we talk about. So it's really important to understand that RNA has the ability to catalyze reactions as well as store genetic information, which makes it pretty unique. No other macromolecule we've talked about has that ability. And this is just talking once again about how we can store that information as DNA. So it can carry that code, just like we talked about with the mRNA. And there's really no difference, except for the small differences we talked about with the sugar and the bases and single-stranded versus double-stranded. But look at flu viruses. Flu viruses carry only RNA as their genetic information. And so it's really interesting that potentially RNA was the starting macromolecule of life, and that this is where everything's come from. And so as we keep saying is that it may be older than DNA, which is kind of a shocker because we think about how DNA had to have been around at the beginning. However, this is only just a hypothesis at the moment. We haven't proven this or anything, but based on the information, it makes sense. So this is the end of this topic lecture. Make sure you understand both topic 1 and topic 2 and how they relate to the central dogma of molecular biology. And make sure you understand really strongly how these processes work, the differences between prokaryotes and eukaryotes, and what the end products are, and how the checkpoints work. The checkpoints are a really big aspect of these chapters for us because a lot of it is review, so make sure you understand those new checkpoints. And let me know if you have any questions.